Thank you for joining us for this session. I'm going to talk a lot about what Bristol Myers is doing to manage some of the challenges we have. And our challenges are interesting because they're not the challenges we would have predicted 10 years ago when the industry thought we had excess capacity everywhere. There were going to be facilities that were mothballed and shuttered and that we would be sitting around with the empty capacity. That's not happening in the industry. We're actually looking all over the world for capacity and we're looking for ways to improve productivity. And I'm going to talk to you about how our portfolio has changed and what we're doing to address that. Before I start though, we have a program at Bristol Myers called Who Are You Working For? And we work for our patients, and so we tell stories about our patients. And I want to tell you briefly about a woman that I know from Indiana from my time there. 2 years ago, this woman was diagnosed with malignant melanoma, and it had spread rapidly throughout her body. She was a friend of ours. She had visited us in Ireland. She's part of this older group of ladies that are kind of semi-retired and go and have fun. They bike, they kayak, they travel. And she was treated here in Boston with a combination of a PD-1 and a CTLA-4, and she responded remarkably well. She had a massive clearance of her lesions. She is not out of the woods. She's still fighting malignant melanoma every day. But I am working for her and for the other patients that are being treated by these drugs. I'm very proud of them. So this slide, which you've already seen, but I'm going to take you down this road, shows what's changing. And what's changing is the number of biologics that are coming to the market every year. And these are just a few. I mean, we just grabbed some of the brand names off the websites, but you can see there have been a number of approvals. There's been 23 new biologics over the last 2 years, and they're still coming this year. And these are not Me Too biologics. These are not another anti-TNF or another anti-VEGF necessarily. These are things that are completely hitting new targets. They're hitting new pathways. In some cases, they may be hitting other parts of pathways, and they're changing the way we're treating diseases. Cancer is changing rapidly. I was at ASCO in Chicago this year, and the patient advocates were ecstatic by the number of new drugs they had to try on the patients that were facing their diseases. We have not cured cancer by any means, but cancer and certain cancers are becoming much more treatable. But we're also still working in the immune system. We're managing autoimmune and immune diseases, and then there's significant metabolics that are coming along as well. So an enormous amount of work. Again, recombinant proteins primarily driven by the antibodies, no doubt, and the antibodies are really coming into their own. So it's an enormous amount. The FDA is swamped with these applications. They're swamped with the INDs. They're swamped with the BLAs, and global health authorities are being swamped by the number of new biologics. But they're also being swamped by the cost. So if you haven't been paying attention to election rhetoric, and hopefully most of us are at this point starting to get a little numb to the rhetoric, everyone who's running for office says that drugs are expensive, and we know that. And it's expensive to bring innovation to the market, and it's expensive to treat these patients. And it's our job to make sure that we're able to bring innovation in the face of the headwinds. So it's really a period of unprecedented growth. Biosimilars are coming. They have been approved in major markets. The scientists in this room and your colleagues at home know how to make proteins. We know how to analyze proteins, and we know how to manipulate our processes so that proteins have certain characteristics. So the biosimilar industry is happening and it's growing. The clinical pipelines are very, very large. They're full, and we're having to make decisions. And then we have big volume drivers. The PD-1s are volume drivers. PCSK9, there's a lot of question around the cost of such a therapy, but it's unquestionable that the volumes are quite large. Not just because we have ineffective processes, because some of these diseases treat millions and millions of patients. And so the kilogram requirement becomes quite large. The Alzheimer's molecules, assuming some of these antibodies actually work, and someday we will be able to tackle these neurodegenerative diseases, but the antibodies that are currently in the clinic have high doses and there are many patients, and they drive enormous volumes. And what we have to do is we have to offset that. We have to offset that by process improvements and making sure that we can make these things more efficiently and more productively. Also to treat diseases that currently aren't treated by biologics and in geographies that do not have the funds to fund large and expensive drugs. So there are companies working on trying to drive the cost down. There are foundations trying to get drugs to underserved people, and I think it's our duty as an industry to continue to drive our productivity, and bioprocessing people are center to that. So 77% of these are in mammalian systems, and this graph kind of shows the volume in terms of kilograms needed, and then this shows the additional clinical volumes, plus just the baseline of making clinical trial material. I mean, most of us are involved in development in one way or another, and so to make clinical material, you have to have a very productive system, because clinical trials are expensive. So this is a snapshot of our portfolio. It probably wouldn't surprise anyone that it's heavily weighted towards immunology and immuno-oncology. But we did an analysis recently, and over the period of about 18 months, we had 9 new compounds in biologics enter our group, most of which were immuno-oncology compounds. And these are the ones you can pull off of our website. These are not the ones that we haven't disclosed, and they aren't ones that are currently under investigation as potential in-licensed assets. So it's an enormous amount of antibodies and antibody-like molecules. There are recombinant proteins scattered throughout. They are primarily expressed in mammalian cells, but there are a handful that are expressed in microbial systems. Microbial systems are still very attractive for some reasons. They're for non-glycosylated or for very high volume products. Microbial systems can really add some advantages. But this just shows you the immuno-oncology portfolio, all of the green are IO. Opdivo is obviously our flagship product. Was launched in 2014, has many indications now across a number of tumor types and continues to be investigated as we learn about tumors' responsiveness to immuno-oncology. But what we're learning is the tumors that are not responsive might be responsive if you manipulate either their microenvironment or the immune system itself. So we and others are investigating compounds like GITR, like CSF1 receptor, the antibody CSF1 receptor, to try to manipulate this immune response and turn what we call cold tumors hot and get tumors to respond. But the combinations of the current ones we have on the market, like PD-1s and CTLA-4, show remarkable results in the clinic, as attested by my friend's response here at Dana-Farber. So it's very exciting. It's almost intimidating sometimes to realize that there's a wave coming, and we have to deliver as a group, as development and manufacturing scientists, and as an industry, because we don't really know which ones are going to work, and we don't know how to place our bets. Significant work still in the immunoregulatory area, the CD40 ligands. And then we have some other interesting kind of non-traditional eTau as a neurodegenerative target, along with A-beta for others. Pentraxin for fibrosis, we've moved into fibrosis, and then other non-immuno-oncology compounds. So quite a bit of variety. What we have done as a company is we've divested of certain areas. We've divested of diabetes. We've divested of antivirals and infectious diseases. Other companies make different decisions, but when you have to focus your resources, you focus on the area where you feel like you can make significant inroads. And I think you'll see more of that in the industry, where people play to their strengths and decide on how they're going to best achieve success in the areas that they're in, which I think leaves room for innovation in all the other areas. There's plenty of innovation left out there. So this is what we face in late-stage development, which is an enormous amount of demand uncertainty. So we've taken some examples, and we do a lot of scenario plannings. We do Monte Carlo models and Excel models, and we have different models for how much material you need to make. But you can see demand uncertainty where you've got a low case and a high case that are twofold different. And we're not talking about, maybe 100 kg versus 200 kg. We're talking about hundreds and thousands of kilograms of antibody or protein, and then a base case that's right in the middle. But if you plan for the lower base case and your marketing people and your salespeople are enormously successful, you're going to run out of drug. And if the case studies of companies that have run out of drug and haven't been able to meet the market are pretty sobering. Some of the first anti-TNF launched was not able to meet market demand, and it's a great drug, but the company that founded it was purchased. And so the failure mode for manufacturing is to not be able to supply the market. The other failure mode is to build facilities that you don't need, but your CEO would rather have an empty facility than empty shelves in the pharmacy. The other drug, this is a 5-fold uncertainty in the forecast. So it's even worse. I mean, do you build Do you build giant stainless steel plants? Do you build a whole bunch of single-use, high-productivity reactors? What do you do to meet this uncertainty? And then what do you do when you start mixing them? Because you're bringing multiple drugs to the market. So how do you start taking that and doing a multifactorial experiment around your uncertainty? DOE with manufacturing capacity. So this is what we face, and so our job as scientists is to deliver really processes and technologies that can match this demand and be flexible, be able to move to different sites. So this is another example of a scenario. So this is showing internal capacity. I'm not using any axes here, but you can see we're bringing on capacity, and I'll talk a little bit about that. So the blue is internal capacity, where we are today and where we will be in the future. The green is locked up CMO capacity, so we have contracts for that capacity. But remember, if you have a contract, you have to pay the contract whether you need it or not. And those of you who work for CMOs, and I did for a while, you know that your clients, when they reserve your facility time, then it's theirs, it's bespoke. And so you need to manage your uncertainty as a CMO, and you do that by signing contracts. And so you get to take or pay on that capacity. So if you look, in some cases, you might not need it, but you got to pay for it anyway. And then you have capacity gaps. They're theoretical gaps. I mean, you're not really sitting there without any material, but most ethical manufacturers need safety stock. You need to be able to take it to all the world geographies. You need to have it approved in all the countries. Every country you may have a change, and then if that's not approved in one country, then that stock can't go to that country. So your ability to move drug throughout the world is limited by both your regulatory and your supply chain. So the uncertainty is compounded on top of that. Now, it is risk adjusted, and we can manage these gaps. You get clever with inventory, you manage drug product at different regions, you work with your commercial colleagues, but every country that you sell drug in, your salespeople in that country want to sell drug, because that's what they get paid for. And so they'll come back and say, look, I need to get approved in my country, and you say, well, we don't really have enough drug to give you, your CEO is going to get really mad. So that's our job, is to manage all of this uncertainty. Now, what we have been able to do, and I think many of you are familiar with this, is drive productivity. And so this is an example of movement from a pretty common cell culture cell line system, DG44. It's very available. Everybody knows it. We know its strengths and its weaknesses, but there are a whole lot of approved drugs that are in DG44. And it can be productive if you push it. In this case, the early cell lines were not particularly productive. They were okay. You were able to make clinical material with them, but you're not going to manage a metric ton demand uncertainty if you're at 2 grams per liter or 1 gram per liter without building, hundreds of thousands of liters of capacity. And so we made the decision to move out of that platform and move to a new platform. And we're on a learning curve. It's a nice curve. It's going the correct direction. So you can see the titers are coming up over time. The latest, and these aren't the latest, these are just some slides that were given to me by the team saying, look where we are and look where we've come. But running in the 5 gram per liter just right out of the gate, that's unoptimized. So that's the new, get a cell line, get it into a reactor, and you're getting into the 5s. And I'm sure you're going to hear more from Biogen and others. You can drive that much higher with clever cell culture work, media development, and working with high-intensity engineering in terms of how you do your cell culture. I'll talk a little bit about that. But even our clone screening has gotten just much more sophisticated, and we can find good clones and better than we could in the past. So that helps, right? I mean, it's a lot easier. If somebody orders 14 kilograms, it's a lot easier to make it a 5 grams per liter than it is for 1 or 2 grams per liter. So that's positive movement there. I'm pretty happy with that. But the other area where R&D really cares is how quickly can you get from DNA to the bedside. And it seems like we've been talking about this a long time, but I'll tell you, it's much harder than most people think to go from the idea of, yes, I think I want to test this, to get it into the patient in whichever clinical setting you're doing. So because there's a lot of changes that we make, and we're scientists. So R&D really likes their production platform. They're using a transient system, and they're making a few milligrams, and they like that. But now that vector doesn't work in the manufacturing system. So you have to reclone the gene, and you have to resequence it. So there's a lot of inefficiencies that we're automatically building into the system, almost by default, without even thinking about it. So what we've done is gone back and said, look, if you really want to move quickly, we have to work together. So we're using the same vectors in R&D that we would use in development, the same methods essentially. They're not all the same, but they at least try the analytical methods that we're using. So we're not completely redeveloping the method. Now, Abby's here. We do have to redevelop a lot of methods, no doubt about it. But a lot of time, that's not our fault. But we have to move more quickly at this. And the other thing we do is we actually move more forward quickly, and I'll talk a bit about that. We also put in disposable reactors. John talked about this. So we make small scale, 50, 100, 200 liter tanks at each of the sites where they can quickly make material for toxicology, for method development, formulation development. And you don't always have to use a clonal cell line to do that. You can do it with cells that are in process. As long as they're making antibody that's analytically comparable, you can start using that reagent very early. You got to move forward quickly into tox, you got to get drug product out, and we had to really shorten the time. We were kind of moving at a leisurely pace before, and I'll show you some examples of that, but we had to speed all of that up and work much more closely and linked to get this to the clinic more rapidly. So this is an example timeline. So I started in 2012. This would have been maybe a median timeline for what we were doing. And so you start with selecting your lead. And once you had a lead saying, okay, well, we think this is the one we want to take to the clinic, we're starting to make cell lines. And then you get the biology. So we have the chemistry, making the protein, and then we have the biology, does it work? And we select a candidate. And then we start moving forward. We get research cell banks, and we make a master cell bank. And then about then, PD would start working on it and say, okay, well, let's see, what does it bind protein A, how well, or if it's not an antibody, et cetera. And then analytical would start working. And then a little bit later, formulation would start working on it. They'd say, well, wait till we have representative material, and then we'll do formulation work. And then we had to figure out what to put the tox material in and then make the tox material while we were doing the human formulation. Then we do the tox study. We'd start manufacturing the drug substance. We'd get that all done, package it up, and then we'd release it, and then we'd ship it to the drug product site, and then they'd get it on their schedule, and we'd start stability, and then we'd file the IND. And it was taking about 24 months at a median, just at a moderate pace. Some of them took longer. Some of them took 36 months. Very few took shorter. That was just the normal pace of development. And so we had to go back and really dig into this. There's some uncompressible things, right? Most mammalian cells take about 20 hours to 30 hours to double. Most, you get a clone, and you can just count how many hours it's going to take to get a 500-liter reactor. Just look at the doubling time, right? There are some things you have to do. Stability is not compressible. So if you need real-time stability, you got to wait real time. But you can go with a small amount of real-time stability data. There is some process development needed, but it's really to check to see if your assumptions are correct. So the first thing we did is we started with multiple sequences. We can't wait till they choose the final. If we do, we're already behind. So we say, we'll start with multiple ones. You give us 4, 6, maybe 8, usually 4 to 6, but you have to choose. So we can't carry 4 to 6 very long, but as soon as you choose, we can start moving quickly. And that gets us a big head start. So the non-compressible parts moved up. The other thing we did is we started saying, look, R&D, you can run columns. Why don't you see if it fits the platform? And if it does, give it to us, and we'll do the final platform fit assessment. We have some high throughput systems. We have some very, very nice scale down models. We'll quickly check that it fits the platform. If it doesn't, we'll tweak it a little bit, and we'll move forward. And we're not going to wait on analytical. We're going to start with the material we have available. We're going to check to make sure that our platform methods fit. Sometimes they don't, but you might know how to tweak it by then, right? So really moved all of that forward. The other thing is we started doing formulation work much earlier. You can take, it's pretty representative, right? What's the odds that the odds are going to be a significant change in stability of a drug product that early? And so we moved our formulation work. And that enabled us to get the tox material out really fast. Almost immediately after they select the candidate, we can almost ship tox material. Sometimes it's a matter of weeks. It just depends on timing. And that pulls everything in faster. It pulls drug substance manufacturing, and we do conditional release for drug product. Because I worked at a CMO where there was a drug substance and a drug product filling line in the same facility. You didn't have to do a lot of release. You just made sure you weren't contaminating drug product. You just move it down the hall. Well, we have our own internal facilities. We just had to get alignment between the two facilities. Yeah, we'll do a minimal amount of testing to make sure we aren't taking undue risk, and we'll start to fill. And that really sped everything up enormously. And then, believe it or not, The people writing the IND were taking their time. I mean, they just had a lot of time because they had time. So, they were going to write the reports and then revise it, and then there's this whole cycle it had to go through. So there's been a lot of work with regulatory and writing to say, okay, we've got the data, let's plop it in and get going. And so at this point, we've really moved it up enormously. And some of these timelines have gotten very, very, very short. Short and easily comparable to a small molecule, easily comparable to a small molecule. So that's good. And it's important because this is some of the innovative clinical work we're doing, and we presented this at ASCO, so I can talk about it a little bit. But we realized that we don't know exactly which of these compounds work with the other ones, right? We do know Opdivo pretty well. We have a good idea how PD-1s work. We have a good idea how PD-L1 expression is correlated with this activity, but we don't know what other combinations are going to drive this efficacy. And so we're doing some really innovative clinical trials where we're doing multiple combinations in the clinical trial to try to really tease out which of these molecules are going to work best together, either with Opdivo as the backbone or some of our other many IO assets to see which ones are going to work. And to get the clinicians to do that, they need the drug and they need all of them. They don't need one and then 24 months later another one and then another one. They want them all right now. I mean, they literally want basically a pantry full of IO assets that they can do work with. And the clinical trial designs continue to evolve as they start to understand tumor biology and biopsies and markers and things like that. So it gives us a lot of ability to do biology in the clinical setting, but it drives us as manufacturers to make sure that the drug is ready. And that's why we've had to really change our tune completely with that number of assets coming in a short period of time. So I think innovation in clinical trial design and innovation in exploratory medicine is going to be critical for really, really moving these forward quickly and finding out what's going to best serve our patients. So how do we assess for platform fit? Pretty quickly, you can run some bioreactor work. We've got some in-process stability work we do, just a quick harvest capture, polish, and TFF. It doesn't take that long to do the in-process stability. You just have to check to make sure the intermediates aren't degrading. They're not going to sit around very long. I mean, if your manufacturing is efficient, get your formulation done and your analytical. The analytical package is your standard package. We don't have, at that point, a really strong cell-based bioassay. Typically, we're relying on binding data or some other surrogate data. And then some quick formulation studies, some thermal stress, freeze-thaw studies, and a slow freeze study to match what you would do when you actually store the drug. Now, we have found occasionally that drugs aren't always stable, or we do see some unusual quality attributes. You can go in liquid formulation or you can go with lyo formulation. If you go with lyophilized, and most people have capability to lyo, you've kind of kicked the can down the road. You're going to have to come back and address it, and you're going to have to do a lyo to liquid transition if you expect to sell a liquid drug. But to get you in the clinic quickly, lyophilization can really absolve a lot of those sins of early phase development. So you'll have to address it later, but it does help. So it's just a quick platform fit assessment. Shake hands with R&D. They agree to it. You work in your own area to make sure you can do this quickly. And high throughput matters. These high throughput cell culture and purification systems are enormously beneficial. And we've been implementing more and more of those as we go forward, both in clone selection as well as in screening studies. And most of our sites have at least one, if not two, high throughput systems available. So the other thing we run into, though, is we get these acquisition things. So we buy a molecule from a small company, and guess what? They have a really bad cell line. It's like you've never heard of it, or maybe you've heard of it, or maybe there's a big royalty payment, or maybe there's just something you don't want about it, or it only makes a gram or a half a gram per liter because it got them in the clinic. So you got to go back, make a new cell line. You got to put it in your platform, our platform. I mean, we could run theirs, but we got an antibody not too long ago. It was a 4-column process for an antibody. Like, well, we were trying to get rid of this one thing. Yeah, but most of us run 2 or 3 column processes, and most of us have those sitting ready to go. So it's an enormous amount of work to bring in a new compound and get it into your platform. And in the meantime, the clinicians are saying, hey, if it works in phase 1, I want to go to phase 3 right now. And then, of course, you want it in your platform for phase 3. You've got comparability challenges. You've got to deal with the FDA or the other global health authorities. So that's where actually I think a lot of our challenges are starting to come from, is bringing in compounds that are non-platform and trying to get them ready for registrational manufacturing. And I know my other colleagues at the other companies are facing the same problem. So one of the things we've done is we've done some network expansions. This 2012, when I started, we had Syracuse, New York, which does manufacturing, but also did development. Devens, it's about 45 minutes from here. We were only doing large-scale manufacturing, and Hopewell was doing small-scale. Bloomsbury is an old Medarex site, and they were doing process and cell line development. And then Zymogenetics was doing process development. A drug product is in Manatee. We've done a lot of network changes. So Hopewell and Bloomsbury are combining into one site that does both early development and manufacturing. Devens, we've added some really nice new facilities. So 2 brand new buildings have gone up and opened up in the last year. Development and small-scale clinical production, plastic, disposable, as well as the large scale. So there's about 200 more scientists now working there than were there before. We've added another manufacturing facility outside of Seattle at Bothell to do small, medium-scale mammalian, and we've also are bringing on a new plant in Ireland. So Devens has been a wonderful expansion. We have a state-of-the-art lab building. We do have 2 trains of disposable capacity. One's built out and one's a shell space. And we can really move stuff through there very quickly. About $250 million investment there. The Ireland plant is going to be a very large-scale facility. It's kind of the medium-sized 6-pack, 15,000-liter reactors, 2 harvest suites, multiple. We have 1 purification train right now with the space for a second, and it'll be a multi-product facility. So again, managing some of that demand uncertainty that I showed you with both small-scale disposable here in Massachusetts and large-scale stainless steel in Ireland. And then we're now renovating the Hopewell facility. It's pretty old. We bought it from ExxonMobil long before I was at BMS, and I can tell you that petroleum labs are not nice biotech labs. So we are renovating those labs and putting in modern workspaces and we're moving our cell line development equipment there, and that'll all be consolidated into Hopewell. And then finally, we brought on a new disposable plant in Bothell. And we've actually moved quite a bit of our clinical production there. It also has a small microbial reactor, so they can cover the microbial portfolio up to a point, and then we outsource it beyond that. But that's given us an enormous relief in capacity. So to kind of wrap it up, we're moving to next-gen processing as well. So really, and I'm sure Biogen will talk more about this, Greg will talk about it, but if you get you want high-titer processes, you have to have high cell density, reasonably high cell density, high specific productivity, and you have to start shortening your cycle time. You really do. But you can get way beyond, the 2, 3, 5 gram per liter. You can get to the 8, 10 gram per liter as you start to do these things. Now, you have other issues. You have downstream constraints that you're going to have to manage as you've got so much protein in a tank. But that just takes good and not completely innovative. We've been talking about this in downstream forever, but it's time that we're actually really going to invest in it. And the vendors are investing as well. I walked through downstairs and we all realize the downstream and high throughput downstream is going to become incredibly important to us. So in some way, shape, or form, we're going to need some type of multi-column chromatography because we can't just keep pouring 2-meter protein A columns. GE would love us to do that, but we really can't keep doing that because you're not going to pour a 4-meter or a 6-meter column. You're going to have some throughput system. And then the UFDF with the single pass TFF systems are really helping. So the scale has to shrink. There's a lot of good academic research going on in this and a lot of the chemi labs, and we have to implement these in an engineering solution to this problem as well as the cell-based solution to the problem. So in the future, there's a lot of talk about facilities of the future, but really what you're talking about is modular, easily changed over. So a lot of disposables, a lot of online analytics. So our Devens plant, our large-scale stainless steel plant, is completely automated. It's a paperless plant. Every single step is run through the building automation system. And I have operators that moved from that to the clinical plant where they're doing the plastic, and I said, what's different? And they go, well, we actually have to do something. Like we have to pour something and change a valve because in Devens right now you don't have to do that. It's very powerful for large-scale manufacturing. It lacks flexibility for clinical, and it's a challenge from an automation perspective. So I think as an industry, if we want to continue to automate, we're going to have to up our sophistication in automation. But a flexible plant gives you flexibility. You can move in and out quickly. Now in Hopewell, if we say, oh, we need a few more kilograms of that, they say, that's fine. It's the same process, same resin, same. All it is a different cell bank. Give us the cell line, we'll thaw it, and we'll make the material. And that has opened up an enormous world for us in terms of our clinical and ultimately our commercial manufacturing. So, our future challenges, odd things. So antibody drug conjugates, they're not new to everyone, but when you put a big, complicated synthetic molecule on the side of a big complicated biologics, even though the molecular weight might be smaller, there's a lot of chemistry going on here, and it isn't as simple as it looks in this diagram. If any of you work on ADCs know, that thing can stick all over the place. Even with the site-specific ADC, you're dealing with some really strange chemistries. bispecific and multimodal antibodies, and now we're all kind of wading back into gene therapy. It's where I started my career in process development, but it's come a long way, and it's real. We're seeing real patient effect for a lot of these therapies. I was just talking to one of my colleagues about that right before the talk, and we have to figure out how to make these effectively and efficiently. And again, they're going to be very expensive. And so you don't want to drop a little bit of that on the floor, and you certainly don't want to leave some behind on a filter. So we've got a lot of challenges from a processing perspective to make these high potency new products. So I like to see people's faces. These are the members of the team that contributed slides and data, though I have a much larger team than this. Some of them are in the audience. But ZJ, Henrik, and Reb were all on my staff, as well as Amara. Reb runs New Jersey. Henrik did run Seattle. He's moved to MS&T, and ZJ runs New Jersey. Lynn's one, and Lee are both two of our project leaders, and Joe does all of our manufacturing scenario analysis. But really, it's a team effort. It's a wonderful team. It's an exciting time to be in our industry. We're having an enormous impact on patients' lives, but it's incumbent on us to make sure that we're doing this as productively as possible. Thank you.